I started my career as a computer scientist, and I avoided biology like a plague. But look at me, right? So life has an interesting way of taking you to the point that you have to go, right? So I started as an engineer trying to build intelligent systems and intelligent algorithms. And that's what my PhD was, trying to make an electronic nose. Turned out it was a very humbling experience. Okay? So as a graduate student, you're cocky, think you can make everything work. You can be the smartest computer program out there. Turned out we couldn't do what the insects can do. So at the end of my postdoc, I realized if I really have to understand intelligence and make intelligent devices, I need to understand the biological basis of intelligence. So I did a postdoctoral work at the National Institutes of Health, working with insects, implanting electrodes in different parts of the brain while they were smelling to understand what is going on here. What are the design principles, computing principles, and so on. And that's basically what we do, a little bit of chemical sensing, both on the artificial side, making an electronic nose, which was initially funded by Dan Prono and Mike Schlesinger. And then now I'm trying to hijack. So if you know that the biology is really good at something, it's difficult to beat biology. It has evolutionary time to perfect its solution. So the goal here is to try to see, let the biology do the hard work of transducing the chemical cue into an electrical signal. And once it comes to the electrical signal, we are really good at manipulating that particular domain. So take the signal off of the insect brain and try to understand what chemicals are present in its environment. So that's the kind of approach that we are trying to take. But for that to happen, we need to understand the basic biology of things, how these insects work, what the anatomy is, what the physiology is, and what are the behavioral aspects of things. And from there, once building on that particular foundation, then we can go into making these biorobots, where we are trying to make new tools for chemical sensing purposes, of course. All right? So let me make sure this works. All right, so this is the talk outline. So I'll tell you a little bit about the kind of problems that we work in my lab, and then get into olfaction, all right? So vertebrate olfaction. How does the mammalian olfaction work? And then get into insect olfaction, spend quite a bit of time explaining the anatomy, physiology, and behavior. And then finally, I'll get into this last aspect of things where we are trying to develop tools where we can make a biorobot. So the purpose of having these outlined is twofold. One, to compartmentalize my ideas into distinct modules. And two, for those of you who are polite, wanting to stay here even though you're bored of this topic, you exactly know how far I am to being done. So it's good, win-win in both ways, all right? So let me begin with motivation, right? So the kind of questions that we ask, again, since I'm trained as a computer engineer, are still to understand how a very simple insect brain performs tasks of pattern recognition, all right? And this can be broken down into three important sub-problems or segments. The first one is the design principles. What are the sensors? What are the transducers that you need to convert an external environment into a signal that you can then interpret and understand? The second component is the computational component. So here the goal is to, once it is represented into a format, let's say electrical signal with which you can work, how do you represent it? How do you perform different kinds of pattern recognition so you can filter out what is irrelevant and focus on something that is meaningful? And the last part is the actuation. The third part is the actuation, where the goal is to take these neural signals and to produce a behavior. So in my lab, we study all these three different modules, the sensing module, the computing module, as well as the actuation module. And then finally, if you think you know the parts of the bicycle is, if you know how it works, maybe you should build it to test whether your understanding is correct or not. So that's basically what we do, trying to make biologically inspired sensors as well as algorithms to test our biological understanding as well as to create novel solutions. And the question, of course, is why insects, right? So, of course, my answer, of course, is why not, right? So there are so many of them. They outnumber us. They have lived on this planet longer than we have done. So clearly, they must have gotten something correct. So the goal, again, if you see, they have unmatched sensing capabilities, right? So for example, male moths have been shown to track female moths from a distance as far away as a football field. And they can do single molecule detection. So it's an amazing feat of chemical sensing. So compound eyes in insects can look at wavelengths that we cannot see. They are sensitive to polarized light, which again, we have no clue about. The locust tympanic membrane, again, can transduce infrasonic as well as ultrasonic sounds, as well as it has nonlinear amplification properties, which are now being looked at to create the next generation of hearing aids. And morpho butterflies, when they stretch out their wings, they have this beautiful blue color. Turns out they have no pigments of blue in their wings. So how they achieve this is they have unique nanostructures of specific aspect ratio. So this actually filters out all wavelengths but blue and reflects only the blue light. So when engineers in General Electric tried to replicate these nanostructures in silicon, what they found was these nanostructures can be exquisite thermal sensors, right? So again, we can learn something from biology there too. And materials such as silk has highly unique properties that cannot be matched with anything that's man-made. So we still have a lot we can learn from these insects. And of course, they are small in footprint, they're highly energy efficient. And more importantly, for our purposes, they actually provide you a low-cost option for creating and validating novel tools, right? And that's the reason why we work with insects. Apart from a philosophical argument that if you really want to understand the biological intelligence, you have to start somewhere simple. You need to crawl before you walk, walk before you run. So if we directly dwell into a much more complex systems with a lot more neurons, much more complicated circuits and behavior, maybe it may be much more difficult to understand what's going on, right? So with that motivation, let me get into olfaction. So from now on, it's all about smell, all right? So I'm going to particularly, let's begin with how we smell, how humans smell, right? So odorants are volatile chemicals. They vaporize, they enter your nostrils, go all the way up to the roof of your nose to this particular region, which is in green called the olfactory epithelium, right? So if you look at this on the right, the bottom right, we have the skull of a sheepdog, all right? So you can see the nasal cavity as well as the bony structures. These bony structures are turbinate bones, all right? Imagine throwing a carpet on this particular area, covering all these bony structures. That is the olfactory epithelium, right? So if you take this olfactory epithelium, spread it out onto a planar surface, from what I have heard is that for dogs, for bloodhounds, this olfactory epithelium is of the size of a small personal-sized pizza. For humans, it's roughly about the size of a chocolate chip cookie, right? So the dogs have much more larger surface area. And apart from that, in dogs, you have 1,000 different types of olfactory sensory neurons. In humans, we have about 350 of them. So there's larger area, more neurons, as well as more types of neurons in dogs. But nevertheless, this is basically where the magic happens, where a chemical signal is then transduced into an electrical signal, and this is then sent to the higher centers, labeled here in 2. So this region is called the olfactory bulb. Right? This is at the base of your frontal lobes. And as you can clearly see, 2 steps and you're already in the brain. So possibly the shortest path from the external environment to the brain is through your nose, right? So these olfactory epithelium that I mentioned, if you can think of studying them with neurons, those neurons are olfactory sensory neurons. What I'm showing you here is a scanning electron micrograph of a mouse olfactory epithelium. So the spherical structures that you're seeing is the cell body of the neuron. And you can see there are 2 wires, one going up, this is the apical dendrite, and this terminates into these hair-like structures called cilia. And these cilia are actually bathed in the mucous membrane. Odor molecules have to reach this mucous membrane, traverse this aqueous medium, and then activate olfactory receptors, which are basically proteins present on these cilia. And that's where the magic happens. We still do not know what exactly these olfactory receptors are tuned towards. So what I mean by that is, so in vision, you know there are 4 different types of sensors: red, green, and blue for color, 1 for contrast. 4 different types of sensors, you're done with vision. In olfaction, first of all, we do not know what each one of these sensory neurons do. And second, in humans, you have over 350 different types of sensors. So it's an extremely high dimensional sensory modality compared to vision, right? So anyways, so this chemical signal then is converted to electrical signal, propagates along these dendrites to reach the cell body where the signals are integrated. And then you have another wire, the output wire or the axon, that leaves the cell body, more on the basal segment, and takes the electrical signal to the higher centers in the brain, right? And of course, the discovery of these family of these proteins that encode these olfactory receptors led to Nobel Prizes for Linda Buck and Richard Axel, I think in 2005 or 2006, right? So a more schematic view of the early computational circuits in vertebrate olfaction is actually shown here. So what I'm showing you here, basically, instead of showing you different types of neurons, 350 different types of neurons, I'm just simplifying it to 3 types of neurons: red, green, and blue. Right? And there are multiple copies of each type of neuron. All right? So that's why you have 2 copies of red, 2 copies of blue, and so on. Each odor is not a lock and key kind of a mechanism. So each odor activates some combination of these neurons. So you have a high dimensional combinatorial code activity at the level of olfactory epithelium, right? and these neurons are scattered along the length of the epithelium. However, the output wires, their axons, are amazingly very well sorted such that the neurons of the same type send their output wires, their axons, to the same spot in the next anatomical region. These are spherical structures of neuropil called glomeruli. Right? This is already in the olfactory bulb. And this massive convergence of redundant input is thought to actually help the system average out uncorrelated noise. What I mean by that is that if you take one sensor, there's going to be some noise in that sensor. So if the signal is very weak, you will not be able to detect that particular signal compared to the noise. But if you integrate from a bunch of these sensors, then you average out the noise which is uncorrelated, and you amplify the signal that is correlated, so downstream neurons can have sensitivity to odors to which this individual olfactory sensory neurons need not have. All right, this enhancement of sensitivity. And from here it goes to a circuit comprising of excitatory cells and inhibitory cells. So again, when I say excitatory cells, what this basically means is that when this neuron is responding, it communicates in such a way that it also helps the neuron to which it is talking to also respond. An inhibitory neuron, on the other hand, it communicates with another neuron in such a way that so as to dampen its response or reduce its response. So positive and negative forces, if you wish. So the interaction between these positive and negative forces converts the sensory input into complex patterns of neural activity. I'll give you some examples of this in my talk. And this is then sent to the higher centers such as olfactory cortex, where further processing as well as association with other sensory modalities happen. And there's also massive feedback from the cortex to the bulb. This is a highly simplistic view of the early computational architecture in mammalian or vertebrate olfaction. Right? This is the same whether it's in humans, dogs, mice, doesn't matter. Right? So now let's move on and see how different or similar is the insect olfactory circuit. Right? So let me focus on insect olfaction. So instead of nose, these insects have their two antennae. And between the two antennae, if you remove this cuticle, the exoskeleton, and a little bit of the air sacs, this yellow region here is actually the locus brain, that the part of the brain that processes olfactory cues. Right? So along the length of these antennae, you have several hundreds of thousands of olfactory sensory neurons. In the locust, for example, you have 100,000 olfactory sensory neurons. So it's an extremely large number. And if, again, you do not have the genetic tools to answer this in locusts, but in flies, fruit flies, for example, they have up to 55 to 60 different types of olfactory sensory neurons. So even there's an extremely high dimensional sensory modality. So here again, these neurons, again, are scattered along the length of the antennae. However, their output wires, again, group based on what type of sensor it is. So all the red olfactory sensory neuron send their wires to the same spot in the next anatomical region. The blue one sent to the blue glomeruli, the yellow one to the yellow glomeruli. Very similar in arrangement to the vertebrate olfactory system, right? And this region is called the antennal lobe, equivalent or analogous to the mammalian olfactory bulb. And here you have processing between, again, you have excitatory neurons called the projection neurons. There are about 800 of them. And then there are inhibitory local neurons, about 300 of them, right? And again, local neurons have processes only within the antennal lobe, whereas the projection neurons send their axons out to the higher centers, very similar to the mammalian circuits, right? And here again, there's interaction between the positive and the negative forces, which transform the sensory input into complex patterns, which are then sent to higher centers called mushroom body, where learning and association with other sensory modalities happen. Yeah?
What part of the insect are you pointing to now? Is it the antennas that you're talking about, or the bright spot there?
Right. So okay, maybe I think I'll just go with
the red light is not
right. So the olfactory sensory neurons are present in the antenna, right? So these regions, this is called the antennal lobe, this is called the mushroom bodies. So these are the downstream centers. So this is the signal processing center. This is actually where the learning and the memory happens, right? So this is equivalent to the olfactory bulb in the mammalian system, and this is equivalent to the olfactory cortex, analogous to the olfactory cortex.
It's smelling with its antennas.
It is smelling with its antennas. Any other questions?
What did the colors represent again?
So the colors basically indicate different types of sensory neurons. Okay?
Are they different proteins?
So yeah, so the current dogma is that each olfactory receptor neuron expresses uniquely one particular gene, one particular protein in the mammalian system. In the invertebrate system, it's two proteins. So one that is unique, one that is common across all these olfactory receptor neurons. Together, they form an ion channel. Again, stop me if you don't understand anything. There's no point me talking if you do not, if I'm confusing you somehow, right? So Again, as you can clearly see, it's a very simplistic picture, but it's strikingly similar to the vertebrate olfactory system. So this has led many people to hypothesize that nature apparently has one solution for non-invasive chemical sensing, and it has replicated it in every single system, in every single organism, across species, across phyla. Right? The goal here is to actually use this simple model organism to understand how odors are represented at the level of sensory neurons, what happens to the signal processing in the downstream centers. So that's so far for the anatomy, all right? So right now I'm going to focus on the functional responses to different odorants at the level of sensory neurons in the antenna, all right? So what I'm showing you here is a nice picture of the locus with the two antennae. On the right, you can see the scanning electron micrograph. And you can see the different antennal segments. And within those segments, you can see small structures, all right? So let me take a small patch of this antenna, magnify this further. So here, you can see those small structures better. What you're seeing is a cone-like structure. Those cone-like structures are basically sensory hairs, right? They're called sensillum. Each one is called sensillum, plural sensilla. And the typical arrangement is as follows. So you have the cell bodies of these neurons at the base of these cones. The cones, again, these neurons send their dendrites along the length of the cones. The cones have pores in them to allow odor molecules to enter and activate olfactory receptors. And that's basically where the transduction happens. Oh, perfect, thank you. So you have the cell bodies at the base of those cones, and then you have, much better, right? So they send the dendrites along the length of the cone, and then you have pores on these cones to allow odor molecules to enter and activate olfactory receptor neurons. And that's basically where the chemical signal gets transduced into an electrical signal. That is then integrated at the cell body, and then you have wires that leave from the cell body to the downstream centers, right? And other thing that you would have also noted is that there are different types of sensory hairs, right? So for example, this short and the small and the short one is called sensilla basiconica. That mediates general purpose odor sensing for food odors, right? The pointy one is called sensilla tricardia. Those type of hairs have neurons that are selective to pheromones. So a male locus can identify a female locust, and female locust can identify a male locust. The tall ones are called sensilla chatica, and they're supposed to be mechanosensors. So you can identify the flow direction of the wind, or when the locust is touching something, you can pick that up. And the ones without a prominent apical cone, these are called sensilla siliconica, and here you have sensors for humidity as well as for acid, as well as for different amines, all right? So there are different kinds of sensors doing different kinds of tasks. And this system, the overall task of non-invasive chemical sensing, is divided into subtasks and arranged beautifully, all right? So irrespective of that, irrespective of which type of sensory hair it is, you have the cell bodies of the neurons at the base of these cones, and then they send the dendrites along the length of the cone, all right? So how do we record neural activity while we provide different odorants, all right? So to do that, what we do is the following. What you're seeing here is the antennae underneath an optical microscope, right? So you can clearly see the sensory hairs along the length of the antenna. And over here, you can see the electrode that we're going to bring in and poke one of those sensory hairs, right? So let me play the movie. So it's very similar to, if you want to hear me speak, you put a microphone underneath my mouth, right? So you're picking up vibrations. So here, so to record neural activity, we're going to poke this particular sensory hair with this electrode. And these electrodes are filled with saline solution, right?
What's the diameter of that probe?
So the tip is 1 to 3 microns, and the impedance is about 1 megaohms. So once we poke the base of the sensory hair, now the tip of the electrode is outside the neuron, and we can pick up electrical activity in the vicinity of the electrode, right? So this is called an extracellular recording. So once we place this electrode, this is the kind of signal that you would get, all right? So there is some baseline noise, but when the odor puff is presented, you start picking these spikes. So these spikes are action potentials fired by the neuron. So basically what happens is the inside of the cell, inside of the neuron, is usually negative with respect to the outside. When it is activated, when neuron is activated, the inside of the cell momentarily becomes positive with respect to the outside. So this is called an action potential or the spike, and this is basically how the neurons talk to each other, right? So if you play a movie of this, right? So if you connect these signals to your audio monitor and you hear these signals, So the popping sound actually is action potentials fired by these neurons in response to the stimulation by an odor puff that was delivered to the insect antenna. So we can, of course, look at this, the size as well as the shape of these action potentials to disambiguate which neuron is responding. And once we have done that, then we can assign a particular, we can rasterize them, digitize that particular information, indicating when a particular neuron is responding to a particular odor, how many spikes it fired, and with what pattern it actually fired. Right? Any questions? All right. Yeah.
So back on the picture of the antenna,
Okay.
where you had those segments. Are each one of those segments pretty much the same, or is there cylindrical symmetries where, you know, they sample chemicals all the way around, or what kind of redundancies are built in, or is everything
Right.
individually?
Right. So what happens?
addressable?
So what happens is there are, again, from what we understand, the 4 types of sensory hairs that I showed you, they are kind of symmetrical throughout. They're distributed everywhere. Of course, not uniformly. So in the tips you have more sensory neurons, sensory hairs than the base. And usually what happens also is that when the locusts are smelling something, they wiggle their antennae to create a kind of a plume or a kind of control the dynamics close to the antennae. And what has been also shown for, not for insects, but for crustaceans, in the invertebrates in the sea, when they have a downward flick, it is equivalent to the inhalation. When they have an upward flick, it's equivalent to exhalation.
I guess my question was more like, does every one of those segments have something for like detecting ammonia or
I see.
bananas or apples or?
I
and
see,
then it
I see.
repeats and you build up
So,
redundancy?
so we have not again, we have not eliminated each one of those segments to see if you have sensors still present. But what we can say is that, yes, you have these sensory hairs throughout. There doesn't seem to be that all one particular sensory hair is exclusively focused on one particular segment. But the numbers can vary, of course. So my assumption is that, yes, you will have uniformly distributed it, so to provide redundancy, and in case you have, also to provide fault tolerance in the event that you actually lose half of your antenna, remaining half can function as well. Yeah?
How repeatable are the patterns
Oh.
for each smell?
It's a good question. So I'll get to this in a minute, just a minute.
So this is the live locus that you're, okay, and you pinned it down,
Yep.
you anesthetize, what do you do to it?
So this is a live locus. The antenna is connected to its head. So it is pinned down, but it is immobilized, so it cannot move. So what is being attached to it is attached to a double-sided tape at the base, so it cannot remove the antenna off. And we also put a little bit of dental wax to hold it in place.
In terms of numbing down,
So in terms of numbing down, if you want to numb it down, what we do is we keep it in the fridge for 10 minutes. And that's enough to numb it down. Good question, though. All right, so what happens? So the pattern that I showed you, okay, this is good for one trial. This is good for one trial. Is it repeatable? All right, so let me address that. So what I'm showing you here are the responses recorded, these extracellular responses recorded from 14 different neurons from a single locust antenna, right? So this is the experimental setup. We're recording from this neuron while we're giving an odor puff. So each tick mark here represents an action potential fired by a neuron. And there are 14 blocks corresponding to the 14 neurons. You have 10 trials for each one of those conditions to show repeatability, right? So the color bar indicates when the 4-second puff was delivered to the insect antenna. And what you see here is that for neurons 1 through 5, there is some spontaneous activity even before the odor puff was delivered. But when the odor puff is delivered, again, there's nothing much happens. So I'm going to classify these neurons as non-responsive neurons. They don't care. They don't respond to this particular stimulus. If you compare that with the neurons 6 and 7, you see that there is some minimal baseline activity. But when the stimulus is presented, the density of activity increases. And it lasts only for about a second and a half after the onset of the stimulus, but it happens for every single one of those trials. And then if you look at neuron 9, for example, here the responses actually increase and remain increased for the entire duration of the odor pulse. And if I look at neurons 12 as well as 14, there is some spontaneous activity, but when the odor puff was presented, it's kind of inhibited or blocked. And then after the odor pulse terminates, it again regains its baseline activity. So you have different kinds of responses from different kinds of neurons. Simplistic, but still diverse. And it's highly repeatable across trials. Right? So if I had to do this thought experiments where I look at a specific point in time and I track the set of neurons that are actively conveying information about a stimulus and move this, as I said, see which neurons are active as a function of time, you would notice that the membership to this set would change as a function of time. What this basically means is that the input from the sensory neurons to the following circuits is highly dynamic in nature. Right? So each odor again activates a bunch of different neurons, and they're highly reliable. That's basically the point, take-home message at the level of sensory neurons. So what happens in terms of signal processing that happens downstream in the antennal lobe? These are the central neurons in the brain. All right? So here we know a lot more, and I can show you. I'll show you different patterns of activity here. So this is again the experimental setup. We have opened up the cuticle, exposed the brain, implanted electrodes, while we are also delivering an odor puff. All right? So we are exclusively going to focus on these projection neurons because that is the neuron that actually integrates information from multiple copies of the same type of sensory neuron, as well as that is the only information that gets transmitted to the following circuits. Yeah.
You may have said it and you may have missed it. When you say an odor puff, are you delivering one single chemical or is it a mixture?
So right now it's all going to be single chemical, but I'll show you mixtures of chemicals as well. Okay.
How do you control the amount that you put in? Is there a certain concentration level?
So what we do is we control the dilution of the odor solution and the headspace. We take a small calculated amount of that headspace and inject it into a carrier stream that goes past the antenna. So what I'm showing you here is what is called as an intracellular recording. So here the tip of the electrode is not outside the cell but inside the cell. And I'm plotting the membrane potential fluctuation, the potential difference between inside of the cell and outside of the cell as a function of time for 7 different neurons in the locus brain, right? And this particular x-axis is time. This particular block tells you when a 1-second puff of hexanol was delivered to the insect antenna. And what you immediately see is that there are, again, these are the spikes that I mentioned about. This is how the neurons talk to each other. And when the odor puff is presented, you can clearly see there is much more complexity in activation patterns. Some are excited and then inhibited and then excited again. And then some are, there's a delayed inhibition, there's inhibition, excitation, inhibition. And then there is a prolonged inhibition followed by excitation. You have different combination of these kinds of responses. So much more complex than what you see at the level of individual sensory neurons, right? And these response patterns are highly odor specific. So what I mean by that is, so if I take a response of a single projection neuron, all right? So this is again a raw intracellular trace showing the membrane potential fluctuation before the odor is presented as well as when the odor is presented, right? This is trial 1. So if I repeat this again for the next trial, I'm just telling you when the neuron is firing action potentials, right? So you can clearly see that this pattern is highly preserved. I can repeat this again multiple times. And what you see here is that the pattern with which this particular neuron responds to this stimulus is highly preserved, highly reliable across trials, right? So what happens if I present a different odorant? So if I present a different odorant, again, you can see that this particular neuron responds to this odor, but with a completely different pattern of spiking, right? And if you present a third odor, you have a third pattern, highly reliable across different trials. Yeah?
So do you know if the sensing part of the neuron is responding to a new molecule each time it fires, or is it allowing the neuron to fire continuously as long as the molecule is present?
I see. So these neurons are downstream in the insect brain. They do not actually come in contact with the external cue. So they're responding to the sensory input from the neurons upstream, which is the sensory neurons. And the extension of these patterns beyond when the stimulus is presented, again, it has something to do with also the circuit mechanisms that are in place.
So, but maybe the question is similar though. Is that a new response or is that a continuing response?
So you're saying whether this response is continuing response or it's a new response?
You have spikes, but is that a continuing response or is that a new response?
So so again, let me understand the question correctly. So are you saying whether these responses are to a different stimulus or to the same stimulus, or?
Yeah, no, same stimulant, but
right.
The point is the same type of stimulant.
Right.
But the question is whether you've turned it on once and it keeps firing, or whether you turn it on and it fires once and then it has to be swept and the stimulant go away and turn on again.
I see.
And it fires and turn on again.
I see. So again, some of these the inhibition that you're seeing is not because, again, I showed you the response of the sensory neurons, right? So sensory neurons, this kind of multiphasic pattern do not happen. So those inputs are either on or off, and they happen, that's it. So here, this particular patterning of responses is mainly due to the interaction between the projection neurons and the local neurons. I'm not showing you the local neurons, so you're not seeing what is the inhibition this particular neuron receives. So it's a result of processing that happens. So it is still responding to the same stimulus.
I think he's asking about out in the sensory neuron in the antenna, about the ligand receptor interactions.
See, so okay, so maybe correct me if I'm wrong. So you're asking about whether once a ligand binds to the receptor, does it get unbound and gets bound again? So I see. So what we know about that particular interaction is very limited. So there are multiple kinds of proteins that actually engage and disengage, and whether once a receptor is bound, how fast can it be unbound? I don't know.
Yeah, what's the frequency of response of
So-
a sensor?
So I think that's basically what you're asking, right? So the pKa values, the kinetic, the how much, what is the binding affinity of those two interacting molecules? I do not know that. Yeah.
Can I ask a related question?
Okay.
Do you subject the locus to an odor it's never encountered before?
So
Say something
All right.
it wouldn't have evolved to
okay.
detect that odor, something you could
Okay.
manufacture.
So let me ask you a question back at you. So would TNT count as such an odor?
Yeah.
Okay. So I'll show you some responses later.
Okay.
So again, it's kind of right. So you can do this very simple thought experiments. Let's say you go to an exotic tropical island and you see a fruit you've never seen before. Does it smell to you or not? Most likely, it's going to smell tropical fruit, right? So I think we have this infinite capability. So this is also thought, right? So if in vision, right? So the kind of natural stimuli that you encounter are limited. So if I have to present a white noise, so where your cable goes off, you have pixels turning on and off. Different white noise stimuli are almost indistinguishable. But in olfaction, there's no such thing. Any cocktail of chemical will give you some odor. So in some sense, it's quite different from vision, and also it can generalize us pretty amazingly well.
And do you learn immediately if you smell it once?
Oh, adaptation. Adaptation happens if you actually do multiple times, yes. So those things are circuit mechanisms that happen higher levels. And when you say learning, there are different kinds of learning, whether you're saying innate learning or conditioned learning, they're quite different. I'll again give you some examples. Maybe you can talk at the end of the talk. So anyway, so this is just one neuron. What I'm telling you is that each odor activates roughly about 25 to 40% of all these projection neurons in the antennal lobe. So the question, of course, is how is odor information represented at the level of this population or ensemble of these neurons? First of all, can we collect this activity of these neurons simultaneously? And how is this information represented? So, and I can show you there are two important things that we do. One is called as a multi-unit recording, where we can acquire simultaneously activity from multiple neurons, as well as we do a very simple analysis, which I'll explain to you how, right? So let's focus on the first part of multi-unit recording. So what we do to acquire activity from multiple neurons is to implant a bunch of wires into the neural tissue. Depending upon how far this tissue is from different neurons, you get different signal. And in this way, you can actually disambiguate which neuron is firing when, all right? So that's the idea here. It's very similar to a conference call where you place a microphone. Depending upon how far you are from the microphone as well as your voice signatures, you can disambiguate who is talking when. So the same thing can be done here. So what I'm going to show you here as a movie is actually the implantation of a multi-unit electrode into the insect brain. So on the top, you have the compound eyes. So you can see the antennae here. This yellow region is the olfactory regions, all right? So this is the antennal lobe, this is the mushroom body. So this is a two shanks of electrodes that we are going to bring in and implant it into this antennal lobe. And this silver wire that you're seeing is basically a micro platform that we are used to lift the brain up as well as stabilize the brain, all right? So as you can see here, so the two shanks are now implanted. So this is a kind of a magnified image of that particular electrode array. You can see the two shanks as well as dots, 4 dots, 8 dots on each of those wires. So each one of those dots is basically an electrode pad. We gold plate them to control the impedance, but they are independent wires. So they get implanted, and once you're in the vicinity of a neuron, we can pick up electrical activity from each of those wires, right? Sounds good. So assuming that we can do this kind of an analysis, this kind of recording, what kind of data do we get and how do we process the data set, right? So let me give you an example. So we do a very simple qualitative analysis called trajectory analysis, and the idea is as following. So let's assume, this is again a schematic, let's assume that we are recording from 3 neurons simultaneously, right? And this is my olfactometer turning on and off. These are the responses of the 3 neurons. So to do the trajectory analysis, what we do is we take these responses, first convert them into rasters, basically indicating when each neuron fired an action potential, right? So once we have these rasters, then we can begin to count, we can bin them in non-overlapping time bins, right? So this tells you how many spikes each neuron fired in any given time bin, and also how the pattern of response changed as a function of time. So once we have these analog spike counts, we can begin to plot them in a 3-dimensional space, where x-axis corresponds to spike counts from neuron 1, y-axis corresponds to spike counts from neuron 2, and the third axis for the neuron 3. For any given time bin, again, you will have some combination of these spike counts, which can become a point in this 3-dimensional space. Similarly, in time bin, in the next time bin becomes a different point, different combination of spike counts, different point. And similarly, you can plot every single one of those time bins, right? So once we have plotted these data points, we can link these points in the order of occurrence. So time bin A connects to time bin B, time bin B connects to time bin C, and we get what is called as an odor trajectory, right? So highly simplistic analysis, right? So typically what we do is not record from 3 neurons, however, we record from hundreds of neurons. These are pooled across multiple experiments. So let's say we are, again, for general purpose, let's say we're recording from n neurons pooled across multiple locus. We create an artificial ensemble of neurons by aligning them with respect to their stimulus. To do the trajectory analysis, we follow the exact same steps, right? So what we do is that we define our time bin boundaries. Once we have our time bin boundaries, we can count the number of spikes made by each neuron. So once we have the spike counts, this is again a high dimensional spike count. So to visualize the responses, we use standard dimensionality reduction algorithms, such as nonlinear techniques like local linear embedding and linear techniques like principal component analysis. So this actually helps you reduce the n-dimensional response and allows you to plot them along 3 axes that best captures that information, right? So again, just to show you what kind of result you can expect from this analysis, let's assume this blue bar indicates the time bin boundaries used for computing the n-dimensional spike counts. And let's also assume that this red sphere is the activity, the n-dimensional vector after dimensionality reduction, right? So what happens is before any odor is presented, you have some baseline fluctuation. But as soon as the odor puff is presented, the activity would change drastically and trace a unique path in this 3-dimensional space and return back to baseline after the odor has been terminated, all right? We can link all these points in the order of occurrence to get what I'm going to call an odor trajectory, all right? Just to recapture, so there is some baseline response, baseline fluctuation. As soon as the odor puff is presented, depending upon which neurons are active, we're going to trace a unique trajectory in this 3-dimensional space and return back to baseline after the odor pulse has been terminated. And this basically was what I'm going to call as an odor trajectory, all right?
So going back to that question about dissociation of the odor from the
Right.
ligand,
Right.
you're going back to a state, so apparently you are you're eventually getting back to the initial state. You're
Correct.
dissociating at some point?
Correct. So I think after tens of, what happens again, so this plot, so there's a unique path with which, so let's say this is your on response when the stimulus is present. After you terminate the stimulus, the receptor neurons will go back immediately to their baseline state within a few seconds. Whereas the following centers need not necessarily go back immediately. They take much more longer time because there is some hysteresis depending upon what stimulus was present. So the dissociation happens at different rates, or the memory is removed, flushed out with different rates at different circuits. Okay. So we can, of course, compute these response trajectories for different odors at different concentrations. All right. So what I'm going to show you here are actual data from about 100 neurons, about 120 neurons, projection neurons in the antenna lobe. They are plotted along the 3 local linear embedding axes. All the red trajectories corresponds to exposure to octanol, an alcohol. The green ones corresponds to exposures to hexanol, a different alcohol with 6 carbon chain length. And then the blue ones corresponds to geraniol, which is a terpene, right? So different odors activate different combination of neurons, which translate into trajectories that evolve in different directions. And if you look at any one of these manifolds specifically, let's say all the responses of hexanol, you have 5 different colors corresponding to 5 different concentrations of hexanol. So the shortest one is the weakest concentration. As you increase the concentration, the length as well as the span of these responses increases, right? So what this basically tells you is that the intensity of a response as well as how long, the duration of the response changes with stimulus intensity. So in this way, odor identity and the intensity information are decoupled and represented at the level of population of neurons in the insect brain, right? It's a highly intuitive way of doing things, but it really happens in the insect brain. So this also allows you to ask several fundamental questions, right? So this is basically what we do in our lab. Is this way of looking at data even meaningful, right? When I say meaningful, as an ideal observer, we know this information exists. But does a locust know this information exists? Can we make behavioral predictions and test them, right? And what is odor identity? Is it the entire trajectory, or is it a certain segment of this trajectory that needs to be in place? And how robust are these responses into perturbations such as presence of a competing cue or humidity or flow rate and so on, right? So again, in this talk, I will show you that we can answer some of these questions, right? So let's begin with the first question of, is this way of looking at data meaningful? So what I'm showing you here are response trajectories corresponding to two different odorants, geraniol and citral. So the blue trajectory corresponds to geraniol, the red one corresponds to citral. And as I showed you before, different odors activate different neurons, so trajectories evolve in different directions. The numbers on the plot indicates time since odor onset. So 0.5 is 500 milliseconds after odor onset and so on, right? I'm just showing you the responses for the entire time when the odor was presented to the antenna, right? I'm not showing you how it dissociates in this particular slide. So what happens is that if both these chemicals are presented simultaneously, what I'm going to call as a synchronous mixture of those two chemicals, then the response to the synchronous mixture is shown here in green. So as you can clearly see here, one component, the geraniol response, dominates this particular competition between these two components, right? So the green trajectories are much more aligned with the blue responses. So to behaviorally test this, right? So if geraniol has one behavior, citral has a different behavior. If this analysis is in fact true, then a mixture of geraniol and citral should have a response very similar to geraniol presented alone, right? So we can in fact test this by using a very simple assay. So this is the assay that we use, which is called a T-maze assay, right? So the locust is walking on an elevated T-bar. So this bar actually is a wooden bar. So we can either choose to go this way or this way. So each one of these arms leads to an odor port where we are delivering either the odor solution, which is geraniol or citral, or the solvent in which those odor solutions were diluted, right? And turns out, again, if the locust walks towards the odor source, it's an attractant. If it walks away from the odor source, it's a repellent. And turns out, the rose odor, again, what I'm plotting you is a preference index across multiple locusts. Positive numbers indicates attraction, negative numbers indicates repulsion. What you're seeing here is that the rose odor attracts locust, whereas the lemon odor repels these locusts. So this basically gives you a very good prediction with which we can test, right? So the mixture of those two chemicals, if our way of understanding the population neural responses were indeed correct, should lead to attraction. And we tested this, and this was indeed true. So this way of looking at population responses to understand what is odor percept, how it influences behavior, it may lead to some meaningful behavioral predictions, right? So can the locust identify a specific odor in spite of other competing cues, right? So that's the next question that we are interested in. So for this particular study, we simplified this particular concept of background. I'll tell you how, right? So what I'm showing you here is a response in the locust antennal lobe to isoamyl acetate, which smells like banana to us, right? And this is the response trajectory that you would get. And if you present an almond odor, benzaldehyde, this is the response trajectory that you would get. So you have activate different sets of neurons, so you have different trajectories, right? So now, when these two chemicals are presented simultaneously, a binary mixture of those two chemicals, what we find is that this binary mixture evokes a response that is kind of a linear sum, or some linear combination of the component responses, right? It's different from the previous mixture where one component dominated, right? So now that we know how the individual component evoke responses as well as what responses the mixture have, we can actually do a small trick, right? So we can actually present the almond odor first, wait for 2 seconds, and then present the banana odor. So after the banana odor is presented, this mixture is identical to this mixture. However, this is an asynchronous mixture, meaning that the two components of the mixture are encountered not simultaneously, but with some temporal differences, right? How does the processing change? So what I'm showing you here in black are the neural response trajectories following the introduction of the banana odor, right? So in fact, if you see here, this mixture is exactly the same as the green mixture. However, the processing is quite different. So now the black trajectories is highly overlapping with the red trajectories. What this basically tells you is that the locust has already identified the almond odor. It no longer cares about it, and it actually can segment this particular component from the mixture that was being presented. All right? So it focuses on the novel cue, on the fresher stimulus. Okay? We can get even more greedier. Can this locust do this particular task even more earlier? All right? So what I can do here is, instead of presenting it 2 seconds after the onset of the first stimulus, I can deliver it within 250 milliseconds after the onset of the first stimulus. Right? And what you see here is that the black responses, again, start off very close to the blue responses and dynamically reshape to get pattern match with the red responses, meaning that the locust is still segmenting the first component and trying to get pattern match with isoamyl acetate as if it was presented solitarily. Okay? So it turns out that the system actually is always looking for novel cues in its environment. So when the fresher stimulus comes on, it filters out, it stops what it is doing, and always tracks a newer stimulus. Okay? So we tested this for this particular odor for different latencies with which we introduced the novel odor. We also presented different combination of these background-foreground combinations, and this particular result that I presented is universally true. Right? So here again, we can do the behavioral testing. Right? So we know that they have information about a particular novel fresher stimulus, but does a locust know? Right? So for this particular purpose, we used what is called as a Pavlovian conditioning assay. So in Pavlov's experiment, you ring the bell, feed the dog, right? So exactly same thing here. So we stop these locusts for a day. We don't feed them any grass. They're highly motivated to learn, all right? So we give an odor puff, and then we give them grass reward. So you do it 5 or 6 times, back to back. They learn the association between the stimulus and the reward really well, all right? And once we present the trained stimulus, they open their sensory appendages close to the mouth called maxillary palp in anticipation of the reward. If you present an untrained odor, they really don't care. Okay, so you can say when the locust is recognizing the stimulus that was reinforced, right? So what we can also do is that what we did was we painted this palp with a green paint, so we can track the movement of this palp and assay the behavior with fine temporal precision. Right? So let me show you a video of this. Right? So here you see the locust after it's been trained. You can see the palps that has been painted in green. So here I've filtered the image to just show you the green pixels, which is basically the tip of those palps. Here I'm going to plot the actual position of the palps, and here the distance between those palps, right? So he or she doesn't care about anything until the odor puff comes on, and then you can see that the trained locust is responding in anticipation of the reward. And it keeps its palp open as long as the odor pulse is present, right? After which it kind of returns back to baseline. Okay? So now we can present the same conditioned stimulus with different backgrounds, right? So that's basically what we did. So what I'm showing you here is, again, this is the first experiment just showing you that the response is selective to the conditioned stimulus, in this case hexanol, and there's no response to benzaldehyde, a different odor, all right? So we can also train the locust with a banana odor, and here you can clearly see the response to the banana odor, but no response to the almond odor, all right? So we can present the same trained banana odor in different conditions, exactly matching our physiology, and the locust can pick up that introduction of the banana odor irrespective of what happened before, clearly matching our physiological data sets, right? So clearly you can pick an odor, recognize an odor independent of the background, and you can do it within 250 milliseconds after the onset of a previous cue, right? It's pretty rapid. Okay, so we know we can do background invariance. How about humidity, right? So humidity is a big problem, right? For chemical sensing, humidity is a problem. The smell of coffee should remain same whether you are smelling it here, close to the coast, hot day, cold day, doesn't matter. But we know almost all chemical sensors have problem with humidity. This is true even for the olfactory sensory neurons. Their responses change quite a bit with humidity. I'm not going to show you the response of the sensory neurons, but if you look at the responses of the projection neurons in the antennal lobe, so what I'm showing you here are responses to 4 different odorants: isoamyl acetate, octanol, cyclohexanone, and hexanol. So there are 4 different colors of trajectories. And the solid lines indicates responses to the odor at 0% RH, relative humidity, and the dotted lines indicate responses to the same stimulus at 100% RH. So what this basic result is showing you is that you can clearly see that those trajectories are highly aligned with each other, meaning that the central circuits can filter out humidity information, correct for the response changes that happens at the level of sensory neurons, and correctly represent these information about a specific cue independent of humidity conditions, right? So again, we can test this behaviorally too. We can train the locust with one stimulus, present them in either a 0% RH case or 100% RH case, and they would respond to the trained stimulus no matter how it is presented. Right? So you can do humidity invariance, you can also achieve background invariance, and always look for fresher stimulus. Right? So pretty novel, unique set of capabilities to have if you're building a chemical sensing system. So let me move on to the last part where we're trying to actually now give this locust a backpack and try to make it sense chemicals of interest to us. All right? So what you see here is actually the wires that are going into the antennal lobe of this locust, which is now plugged to this particular chip. All right? So to create this kind of a bio-robot, we need three things. One, capabilities to actually make a minimally invasive surgery so we can implant the electrodes and let the locust recover so we can walk and do its behavior. And you also need this particular chip, a small chip, that low weight chip, that you need to actually collect the data as well as transmit this information wirelessly. And third, some mechanism with which you can steer the locust to the portion, to the region of your interest. Right? So I'll show you that we have made some progress in all these three areas. And in some sense, the philosophy here is actually very similar to what was done when coal miners used to take a small bird, a canary, into the coal mines. So the bird dies, then basically means you have to get the hell out of there. So there, the readout was coarse. Now we are saying we can make much more precise readouts. That's only the difference, right? Philosophically, they're similar. So just to again give you a comparison, right? So this is a BioNose, an antenna, which has about 100,000 sensors, whereas this is a state-of-art chemical sensor that we actually are working with National Institute of Standards and Technology in collaboration to develop this. So here you have basically the transducer is a metal oxide it's a semiconducting metal oxide chemiresistor, which sits on top of an heater. So we heat this metal oxide, expose it to different chemicals, it undergoes a chemical reaction. And if you put different materials, you can get a fingerprint for each odor. And you have 16 different sensors. This is a package device. So this is state-of-art with 16 sensors. What I'm telling you is that the biological nose has 100,000 sensors. So biology can beat engineering, state of engineering, purely based on complexity of the solution, right? So what I'm showing you here, again, let me play a movie here. So what I'm going to show here is a locust which has been implanted with an electrode. And it has been, again, put back together. And it recovers back really quickly. And it begins to walk and jump and fly and do all kinds of things. So this is within 10 minutes after the surgery and implantation of the electrodes. So the wires are still sticking into the antennal lobe, and the locust can move about freely without having any problem, right? And if we connect these wires to an amplifier, we can get spiking responses highly similar to the ones that we see in a highly invasive surgery, right? And what you see here, again, these are 4 different odors being presented. And you can clearly see that the density of the spikes as well as the patterns with which they spike are quite different depending upon which odor is being presented. Okay? So this is all well and good, but can the locust actually have sensitivity to some chemicals that may have some engineering relevance, right? So what we did was we actually tested with TNT vapors. And these are the responses of 4 representative projection neurons in the insect antennal lobe to the TNT vapors delivered at 25 ppb. And what you see here is that some are responding early, some are responding late, even more later, and some are inhibited. So for some reason, these locusts have responses to both TNT as well as DNT. We do not know the ecological meaning to it, but that may be, going back to your question, can the locust pick up signal that it has never encountered before? So clearly it does seem to. And if you look at the responses of about 80 neurons, right, again plotted along the first 3 principal component axes, so all the red responses are to TNT, all the blue responses are to DNT, the green ones are to hexanol, and then the gray ones are to hot air samples. And you can clearly see, depending upon the pattern of activity across these neurons, we can distinguish these different conditions. Yeah.
Concentration difference there between TNT and DNT.
Right, that's because of the differences in vapor pressure.
So you wouldn't be able to sense the 25 ppb if it were DNT? Or
So I think we do not know that yet. So we're not trying to match this. So we just matched it based on the flow rates and the volume of odorant that is being injected. So pretty sure we can dilute it even further and check whether the responses to DNT exist when it goes to 25 ppb. Okay? So nevertheless, this basically tells you that, okay, there is some promise in terms of using these kinds of insects and looking at their responses to certain target chemicals of interest, right? Of course, what we also want to do is to steer these locusts to regions of interest. And here is where my colleague here at Washington University, Srikanth Singamaneni, came up with this idea of giving these locusts a tattoo, a plasmonic tattoo. All right. So this tattoo has two parts. It has gold rods, which of a specific aspect ratio, which converts light into heat. Right. So, and the second part is a silk film which allows these gold nanorods to be attached to any surface, wings, antennae, compound eyes, doesn't matter. So the plan here was that we can shine laser after we implant these locusts with tattoos. So that will provide a local heating, and these locusts do not like the heating, the heat stimuli that is being delivered. So they will get repelled away by this particular stimuli. In this way, you can control the navigation of the locust. Right? So we validated this by actually making the locust again walk on the rod, and when the tattooed locust or shined with a specific laser light, they again get repelled and they walk away quickly. Right? Much more faster than those locusts without the tattoos. So this way we can non-invasively try to control the behavior of the locust. In addition, what he also did was he functionalized these gold nanorods with certain peptides that are known to bind to TNT. So once functionalized in this particular fashion, you can make these locusts walk through a container with TNT vapors, and when you collect the the locust back, if you do a Raman spectroscopic analysis before and after, you can clearly see there is a very nice unique peak at 1350 wave number corresponding to the NO2 stretch. So you can use the same technique to both steer the locust as well as for collecting samples from a region of interest. Again, this work is still in the preliminary phase, but that's basically where we are trying to go. Trying to take advantage of the biological sense of smell by reading off the neural activity, while it does the difficult task of transducing the chemical information into an electrical signal and giving it a backpack so we can record the neural activity as well as transmit it. And in addition, also come up with the ways to steer them to regions of interest as well as collect samples from the regions of interest.
So if you conditioned it to get food reward
Right.
for TNT, wouldn't it just learn to approach where TNT is?
So that, we are looking at that. So yes, we are trying to do that. So hopefully I'll have some answer in a month or so. So that's basically all I have to say for today. So we are looking at the basic olfaction, the design computing principles of the locust olfactory system as well as its behavior, and try to develop engineering tools with which we can hijack their sense of smell. Right? So as my colleagues say, imagination is the limit. Future work is also to try to make transgenic locusts which can actually express genetically encoded calcium indicators across neurons. So we not only collect from few spatially located, spatially proximity neurons, but from neurons throughout the brain, as well as to maybe upregulate or downregulate certain receptors to enhance sensitivity to certain target chemicals. Right? So with that, let me also acknowledge the people who did the work. Devajit Saha, a talented postdoctoral fellow who did most of the electrophysiological recordings. Srinath Nizam Patnam, a PhD student in my lab who did all the analysis. Darshit Mehta, who's involved in developing this backpack. Undergraduate students, Sarah, Ege, Alex, and Ray did a lot of these, contributed in multiple ways to this project. And my collaborators on this project, Srikant Singamaneni, who's a materials expert, as well as Shantanu Chakrabarti, who's an expert on miniaturized electronics. Funding from Office of Naval Research, thanks June, as well as NSF Career Grant. All right. With that, I'll be happy to take any questions you may have.